So today we're going to cover the membrane, the structure of the membrane, transport across the membrane, and signaling between cells, which has to do with, go figure, the membrane. Today is all about the plasma membrane. We talked about the plasma membrane previously. It separates the inside of the cell from the outside of the cell. And we've covered that in depth. It surrounds the cell, but not only does it surround the cell, it surrounds all the compartments of eukaryotic cells. It's good for a bunch of stuff. Selective uptake, cell compartmentalization, protein sorting, anchoring the cytoskeleton, cell signaling, and cellular adhesion. We'll go through each. So if you haven't written them all down yet, don't worry. They're going to pop up at the top of each slide. So first up, selective uptake and export. The cell only wants to bring in certain molecules. It only wants to secrete certain molecules. It's that membrane that acts as a gatekeeper that allows only some things in and allows only some things out. It's vital for compartmentalization. Here's a picture of the cell that I will never make you draw or label.
I thought that cell picture was inaccurate.
It is. It's absolutely inaccurate. But the idea is there are membranes surrounding each of these organelles. Those membranes are made of the cell membrane or plasma membranes. Yes?
So when they were making this, did they just draw what they thought was right?
What they did when they were creating this, as far as why it's so inaccurate, is because they wanted to create an educational view that 5th and 6th graders could understand, which is why it's cartoony. The membrane is important for protein sorting. You can move proteins and polypeptides from one point in a cell to another using these protein sorting pathways with membranes. You'll end up moving from the endoplasmic reticulum to the Golgi apparatus by having a vesicle pop off of the endoplasmic reticulum and fuse to the Golgi. Do you guys remember how that happens? How you get this to start pinching off? What parts of the cytoskeleton are involved in that? You get squeezed tighter and tighter and tighter. The membrane deforms. It squeezes. 3 parts of the membrane. 3 parts of the cytoskeleton. What are they?
Was that the last?
That was the last lecture. There are 3 parts of the cytoskeleton. Microtubules, which act as rails. Actin filaments. Actin, right? Actin causes action. Actin causes the deformation of the membrane. Then we've got to move that vesicle from here to over there. What will it travel along? The railway, the microtubules. But it can't just travel along the railway without some sort of force. What carries it along that railway? Type of protein? Motor proteins. So motor proteins are going to take the vesicle, bring it over to the Golgi where they can then fuse. That's part of the protein sorting pathway. The membrane is also involved in anchoring the cytoskeleton. Again, we just talked about the different pieces, the microtubules, the actin filaments. The last one's the intermediate filaments that provide rigidity, strength, structure. They're the tent poles in the tent. They're going to be anchored in the membrane. So one end is going to be stuck in the membrane, the other sticking out. It's going to be very helpful in cell signaling, bringing a signal from the outside of the cell to the inside of the cell. So here we've got a signal molecule interacting with a protein in the membrane. That will then release a signal on the inside that can cause something to happen. Without cell signaling, your cells cannot coordinate their responses. So before we talk about how we get these different functions, we have to talk about the structure, because in the end, form will always determine function. So let's go over how the membrane gets its form, what it's made of, and how the chemical interactions can lead to a function. So we'll begin at the beginning. A biological membrane is often called a mosaic. You guys ever made mosaics in like an art class? What is a mosaic, generally? Yeah, you get a bunch of little pieces, a bunch of colors, and put them together to make a picture. So here we got a whole bunch of little pieces, tiles, white ones, blue ones, yellow ones, green ones. Individually, they have absolutely no merit. They have no properties associated with them, aside from their color. It's only when you put them together in a certain way that you get what's called an emergent property, something new arising. Biological membranes are very similar. Individual pieces don't do much, but when you put them all together, suddenly new properties arise. Pardon? It's the same with atoms, yeah. There are 3 tiles that make up this biological mosaic, and you've heard of them all, I hope. Lipids, proteins, carbohydrates. These are 3 of our classifications of macromolecules. One of our big classifications is missing. What classification is missing? Starch is a carbohydrate. Waxes are lipids. Nucleic acids are missing. We're missing the nucleic acids. No DNA, no RNA on this outside. But there are lipids, proteins, and carbohydrates. By far, the most abundant molecule in the phospholipid bilayer, or in the plasma membrane, is the phospholipid, which is why it's alternatively called a phospholipid bilayer. Here you can see the phospholipids as these sort of ping-pong balls with tails, and then embedded within them are different proteins. Now, what do you know about the solution that's up here? It's water, so it's an aqueous solution. And down here? It's also it's also water. So we have seen these phospholipids before. They are amphipathic. What does it mean to be amphipathic? Just the opposite, but you're dead on. It's got a polar section and a nonpolar section. These phospholipids in particular have a polar head and a nonpolar tail. That polar head then is going to orient toward water, and the polar tail is away from it. So one part of it's polar, one is not, is amphipathic. For the phospholipid, you definitely have a polar head and then 2 nonpolar tails. And just take a look at the structure of it. You've got, again, polar head and then a long straight tail and then a tail with a kink to it. Now, just like with unsaturated fats, why does it have a kink? That's right, it has a double bond there. And that bends the tail. That's going to come into play in a few minutes. But that's not the only molecule that's in the phospholipid bilayer. There's all sorts of other things. There's also proteins. Now, proteins, remember, are made up of amino acids. Here, we have different proteins embedded in that bilayer. And you'll notice these little squiggly things, these helixes. Where have we seen those before? Protein structure. They're secondary structures. What's that secondary structure called? An alpha, yes, an alpha helix. An A helix, yeah, that's fine. These alpha helices they do have that coil shape to them, and that shape's going to come into play because form determines function. You know that the things that are facing this section right here have what chemical properties? The amino acids that are right in the middle of that phospholipid bilayer have what chemical property associated with them? Let me put it a different way. So this is a phospholipid bilayer, right? These are in this bilayer. So what do we have to know about the chemical properties of it? Okay, it definitely has to deal with water. What does it have to deal with water? It doesn't like it, so it's hydrophobic. And thus up here, where it's exposed to the water, hydrophilic, and down here, hydrophilic. So you have a hydrophobic region that anchors it in that bilayer. These alpha helices serve other points too. You guys have seen, I assume you've seen a slinky before, right? Yeah. Okay. No? Well, sorry. If you've seen a slinky or a spring, it coils around, but you can look through the center of it. You can drop something through the center of it. These end up then becoming pathways through the membrane.
You said that the hydrophobic layer
Anchors the proteins. Yeah, so they can't move once they're in there. They can't move out of the phospholipid bilayer.
but it's still like a pathway
And it still becomes a pathway because it's like a slinky, like a train tunnel going through a mountain. You can't move the tunnel in or out of the mountain. Ooh, tricky. So you've got these proteins embedded in the bilayer, and you'll also have carbohydrates attaching to them through a process called glycosylation. We've heard of glycosylation before. Where did we hear about glycosylation? 10 minutes ago. That's 10 minutes ago when we went over it. So glycosylation happens in what organelle? The endoplasmic reticulum. Glycosylation is the attachment of a carbohydrate. So if we add a carbohydrate, we glycosylate a protein. It's no longer just considered a protein. Now it is a glycoprotein. Because why not? We add a carbohydrate, we'll add glyco to the prefix. If we add a carbohydrate to a lipid, it's called a glycolipid. That's just terminology. Again, we don't have a very clear understanding of why glycosylation occurs. We do have some really good hypotheses I'll talk to you about, but it hasn't been solidified. These membranes are in a constant state of flux. They're constantly shifting around. It's almost like a crowded room. In a crowded room, people are always moving, aren't they? Even if it's not very much, they're shifting around each other. Maybe they're trying to get more relaxed. Whenever there's one of those, like a lockdown drill or something, it's not like you just stay stuck in one spot. People are leaning against a wall. They move. They go to sit near somebody else. There's constant movement. You've witnessed in a stadium of people, thousands of people sitting down. There are emergent properties that happen as people shift and as they move a little bit. You see a wave occur. You've seen people do waves in a stadium, right? That's just a little bit of movement, but there's constant movement. Go to a movie theater. There's still constant movement, even though we're supposed to be sitting in our seats. I know, I just went to the movies this weekend, and people got up like 15 times in front of me. What was it called? It was the...
Jack Black?
Yes, it was. It had Jack Black in it. It was the house with clocks in the walls or something.
Was it good?
I went with a bunch of kids. It was a super fun few hours for me.
Is Jack Black amazing?
Jack Black is usually amazing.
Yeah. That's the right answer.
So,
He's good and like not children.
right. Actually, in this one, he was very good for the part. But you know what? We're going to stay on topic. So, these things are constantly sorry. These things are constantly moving around. Think of it as sorry. I'm going to need a second. I'm sorry. Now, integral membrane proteins are going to be embedded within the membrane. They're stuck in there. They can't move. Why? What's going on right here chemically? Yes, hydrophobic thingies. Well said. There's yes, it's hydrophobic. Yes, we have a hydrophobic effect going on. They are stuck in the membrane. They can't move up. If this was to move upwards, what would happen? It would be in the hydrophobic region. It would be in the hydrophilic. There's water up here. It hates water. It's going to come back down here. It doesn't want to move. It is stuck. Same here. This is called an integral membrane protein. It is a lipid-anchored protein. It's anchored in the bilayer. It's not moving. Do you see? So what we have is a hydrophilic region up here, a hydrophobic, a hydrophilic. Same structure, hydrophilic, hydrophobic, hydrophilic. Hydrophobic. When you've got those hydrophobic bonds, you anchor them solidly into the membrane. They're not leaving the membrane. They can float along the membrane, just like if you were to throw a log into the ocean, part of the log's underwater, but it's sort of still floating. It's moving along. Attached to these integral membrane-bound proteins are termed peripheral proteins. Peripheral proteins are on the outside. Think of your peripheral vision. It's to the sides. So you've got something going on associated with the membrane but not directly connected to it. This is connected to a protein that is connected. So this is in the periphery. Does that make sense? So peripheral membrane proteins, integral membrane proteins. The peripheral ones may be important for cell signaling, because a signal can come in on one side of the membrane, interact with that protein, and cause this one to release on the outside. Yeah. So integral ones are integrated in. They have something, they're connecting with that hydrophobic region. Here, here, here. Peripheral ones are not touching that hydrophobic region. See the difference? Okay. So integral membrane proteins are embedded in the plasma membrane. They're touching the hydrophobic region. Okay. Peripheral proteins, they do not touch the hydrophobic region, but they're still close to the membrane. So this plasma membrane is what we would term a semifluid. And the reason for that is it only moves in two directions. On the plasma membrane, you can move forward and backwards. You can move left and right. It's hard to move up and down. In regular fluids, you can move forward, backwards, left, right, up, and down. When you're swimming, you can dive down, you can go up, you can swim left, right, forward, and backwards. You can't do that here. But there is still constant shifting around. Can I borrow this? Thank you. And yes. So what's happening is you might have, there's your squeaky container again. You might have these shifting around. That's possible. Or if these two are linked together, they have a process that both of them are needed for. For instance, pulling in different types of solutes from the environment to create a new macromolecule inside of the cell, they might need to be linked so they don't move apart. These guys are constantly moving, but they can become linked. The way they can become linked is if they are glycoproteins. What is a glycoprotein again? Protein with a carbohydrate attached. So what can happen is the two carbohydrates can reach out and bind to each other. If you've seen Gilligan's Island, and I don't expect many of you have, but if you have seen it, okay, good, thanks. It's a bunch of castaways stranded on an island. They have rope. I'm sorry, they don't have rope. They have vines and they have bamboo. If you take bamboo and throw it in water and you try and step on it, what happens? It either goes down or it's like splits apart and goes sideways. In order to create a raft, you need to bind them together in some way. In this case, they binded them together with vines, and then they made a raft they could leave the island on. Never worked. But same thing happening here. These would be the bamboo. The carbohydrates attaching would be the vines that link them together. So this is one possible thing, reason we have glycosylation. Yes?
Well, with the raft,
yes.
Is there like a weak bond between them?
Where it all splits apart? There could be, but most cells are evolved in such a way that that doesn't happen. All right, so we have these rafts floating along. The cellular environment, though, is not static. It's changing. The waves, the sea could get rough and turbulent, or it can be calm. You can have a lot of motion or a little bit of motion. So there are a few things that affect fluidity. And it's all about the phospholipids. First off, length matters. The length of the tails of the phospholipids plays an abnormally large role in how fluid the membrane is. The reason for that is it's all about distance. Haley, can I use you for a second? Here, stand up. Hold out your arms. Okay, so grab my hands. We have a lot of motion, right? I can move left and right all over the place. We have less, there's a lot less motion. Does that make sense? Thank you. It's swing dancing versus 8th grade dance. You know, it's like, I assume, I don't know what your 8th grades are like. But for the most part, they do this whole weird zombie thing. So the longer the tails, the longer those connections, the more fluidity there is, the more motion there is. The kinkier a phospholipid is, the more fluid you have. Where that kink is, that bend, that's where you're going to have the more fluid. So depending on where that kink is right there, where that double bond is in the tail, you could increase or decrease fluidity. If the kink is at the very bottom, these phospholipids can get right next to each other. They squeeze in tight and there's not much motion. If the kink is farther up, the tail juts out at a more awkward angle. And when that happens, the two phospholipids are farther apart. So there's more motion they can have. If you've gone to a concert, at the very, very front of the stage, there are people packed in really tightly. And if you haven't, you're just not cool like me. Don't get me wrong. The last concert I went to was like the Teenage Mutant Ninja Turtles concert. So, yeah, 1987. Oh yeah, sorry, I forgot you guys weren't born then. Coming out of our shells. I still have the thing. Vanilla Ice sang at it. What? Was I in the mosh pit? I totally wanted to be. You go down. Actually, I take it back. I went to a They Might Be Giants concert. Again, dating myself though. So when you're in a concert, you can be up front and it's really tightly packed. People are near the stage and they're not moving much. You got that weird swaying thing they're doing? You hit a mosh pit though, what happens? They're a bit tighter together, aren't they? They're tight together until they start punching. It's never be stepped on. So, the more motion you have, the longer your kinks. Think about in a mosh pit, if people are swinging their arms around, you're not going to be too close to them. Or you take them down.
I'd love to see that.
That turtle concert was rough. That's why you have a shell. The cholesterol in the membrane will keep the membrane stable. You guys have heard of cholesterol, right? Cholesterol's bad, except it turns out we kind of need some. Granted, we don't need nearly as much as we take in. What cholesterol does at high temperatures is it holds the membrane together so it doesn't fly apart. At low temperatures, though, cholesterol acts as a lubricant. It allows these phospholipids to slide past each other. So between these three things, length, kinkiness, and cholesterol, you can determine the fluidity of the membrane. Cholesterol holds them together, makes them more stable. It holds them together at high temperature, or it lubricates them at low temperatures so they can slide. I realized as I was going through it with the last class, most of these big concepts all have 3 parts to them. And coincidentally, most of my exams have 3-point questions. I just wanted to remind you, in case something like this pops up on the exam. I have no idea. Probably not, because I'm lazy and I wouldn't want to grade it. That's the right answer, thanks. So, we've got the movement of the membrane now. We've got rafts floating along it, held together. Some of these protein rafts are floating. They're sort of, yes, that's a house in the ocean. Some of these houses are floating. They're moving with the current. Houses. Some of these proteins are floating, moving with the current. This house got washed out to sea. And I don't think it was, I think it was Hugo, actually. You never heard of Hugo. Okay.
Did you say that the semifluid membrane was like it was like the wood that's like sometimes floating up?
Yes, a semifluid is you can move forward, backwards, left, and right. You can't move up and down. Yeah. So like this house, it's not tethered to anything. It can just float along until it eventually sinks, but it could just keep floating. Some proteins are stuck in one place. Buoys don't move much. So here's my question to you is, what would this be stuck to that stops it from moving relative to the inside of the cell? Okay, if this is, sorry, this is a protein? Sorry. This is a protein in the phospholipid bilayer. Sometimes they just float around. They're not anchored to anything. But if they end up stuck in one place, they don't move relative to the inside of the cell. What are they anchored to? Think, okay, boats, right? If you have, I'm going to steal your stuff again. You got two boats. If you link them to it, you tie them up to each other, and they're out in the ocean, they still float along, but they float along together. Does that make sense? That would be what glycosylation is good for. But how do you get them so they stop floating along? You anchor them. What do you anchor them to in the ocean? You anchor them to the bottom, right? It catches something. You've got a very, very heavy chain or a very, very heavy object down there. It's stuck on something. But you can't do that in the cell. So what does it anchor to in the cell? No, it doesn't have to do with the word hydrophobic, sorry. Back to hydrophobic. Well, that's my question. What? Okay, here's your hints. Think about your body, right? You move things around. What keeps your skin in one place? You know what? That's the best answer I've ever heard. My belly button. That's okay. So Bio 102 is next semester.
You're not going to make it.
You're not going to make it. All right. That's exactly what it's anchored to. Thanks for recovering this, Haley. No, we're going to ignore the belly button comment. We're moving on with Haley's correct answer. So, the answer is the cytoskeleton. The actual thing, yes. Actually, this protein has to be anchored to the cytoskeleton. Specifically, trickier. Well, actually, we got to go to hint, so it's one of three answers. Back to threes. What part of the cytoskeleton does it bind to? That's exactly right. Intermediate filaments. Why? Because they give it structure.
Wait, how is that related to the cytoskeleton?
It's the intermediate filaments are a part of the cytoskeleton that this would bind to, are the specific component. The intermediate filament provides rigidity and strength. So the protein will bind to the cytoskeleton. How would it bind? Because it has what kind of region associated with it that allows it to go through the phospholipid bilayer? Hydrophobic region. You were getting to that. No, both you and Amber are like, oh no, it's got to have something to do with that hydrophobic region. Okay. Pardon?
When you said that an intermediate filament provides strength, is that it provides strength so that the protein can anchor to the?
So the protein can anchor, yes. Well, and for a number of other reasons, but that's one reason. Yes?
How do you know which will float and which will be anchored?
Typically, you'll just observe it. So some of them are floating and some are anchored, and you just notice that. Correct. If it's anchored, it's to the cytoskeleton. All right, so now we've gotten to the point where we've got the structure down. If you can understand the structure and the form, the function follows from that. It's made up of phospholipids, proteins, and carbohydrates all working together. The phospholipids, these amphipathic molecules, build a bilayer. There are proteins embedded within that cause things to happen in the cell and outside of the cell. And those can be held together using those carbohydrate strings. Those glycosylated proteins and lipids can connect to each other and form these raft communities. So now let's talk about how we build it, how it's created. So membrane synthesis. Where in the cell does do phospholipids get built? In the cell? Where in the cell? What? You know, it's not the nucleus. Okay, that's one gone. It is the ER. Which one? It's the smooth. So we were going to build these phospholipids in the smooth endoplasmic reticulum in 7 steps. Step number 1, get all the pieces together. You need these enzymes. You need acyl tails, glycerol, phosphate, a polar head group. You either need to build them or you need to pull them in from the outside. Then you have to get all of those parts together. You have to get them together at the smooth endoplasmic reticulum. They need to be in the right place at the right time. All of the parts need to be in the right place at the right time, which is the smooth endoplasmic reticulum. Why is it? Because this was in this is an iconic image of the end of the war, and the photographer was in the right place at the right time to snap that image. That being said, this image says a whole lot because it's supposed to show celebration, and what it shows me is look at her arm. It's not a doll, no. It's a woman. He grabbed her and kissed her. He had no idea who she was. She had no idea who he was. So even though this was that iconic image, now it's evidence of sexual assault. Now she's in her 90s now, but she's like, nope, it was fine. Don't worry about it. Step 3, coenzyme A comes in. Coenzyme A is an important enzyme that does a whole bunch of things in the cell, one of which is to help build phospholipids. It's going to attach to and activate the fatty acid chains. So we've got our fatty acid chains. The coenzyme A is going to come in, bind, and make them ready to bind to other things. Now I bring this up simply because in, about 2 weeks, I'm going to give you guys a lecture on the on cellular respiration. And when I'm going through that, coenzyme A is a really, really, really important enzyme. Like fantastically important. And I'm going to turn to you and say, hey guys, where have we seen coenzyme A before? And then you're going to look blankly at me and throw out a whole bunch of random answers. And I'm going to listen and I'm going to say, man, all right, you guys think about it. We talked about it. I'm going to go get a drink of water and then I'm going to come back in. And then I'm going to do that and come back in. And I'm going to bet it's either going to be Amber or Haley with perhaps Isaac or Chris as a close-up, who's then going to be able to answer it. No, not until Amber says it first. So, and only because you guys tend to be the louder ones. But there we go. So I'm going to do that and give you that heads up. Right now, I'm going to ask that question, so be prepared. I've done that for, 12 semesters now, and that's how it always runs. Next up, we've got our fatty acid tails activated. They're going to have to bind to a phosphate in the glycerin molecule. So there's a glycerol and a phosphate. They bind to it. The fatty acid glycerol phosphate complex is then inserted into the smooth endoplasmic reticulum. So now we've got our nonpolar tails, our hydrophobic tails. So they are going to very easily fit into the membrane. But the problem is it's still got this phosphate head. So we come up with an enzyme called a phosphatase. It means it cuts it. So that enzyme comes through and hacks off the phosphate. Now it's headless. Right, you can't have Voltron without the black line. I go to step 7. Then you have a choline group come along that acts as a head. So the phosphate's gone, the head binds, and now we have a fully completed phospholipid. Okay, so now I'll answer your questions. Start over?
Yeah.
I'll just let you guys catch up with your writing real quick.
You saying the Voltron thing sounds like
There's no words in it.
I know.
So get all the pieces together, get them to the smooth endoplasmic reticulum. activate the fatty acid tails, add the glycerol phosphate head, insert it into the membrane, remove the phosphate, add a choline head, and now we have a completed phospholipid. Any questions? Yes. Step one, we're going to get all these pieces together through ingestion or creation. Then we get them all shipped over to the smooth endoplasmic reticulum. At the smooth endoplasmic reticulum, the tails are activated by coenzyme A. I think we're at step like 3 now. Okay, step 1, get everything together, get everything into the cell. Step 2, get everything to the smooth endoplasmic reticulum. Step 3, activate the tails with coenzyme A. Step 4. Step 4. Attach a glycerol phosphate group. Glycerol, GLYCEROL /PHOSPHATE. Next, insert into the smooth ER, into the membrane. Okay, then remove the phosphate. And then add a choline head. Any questions? Do you want to go over again? You know, I'm not even going to ask you any questions about it, so, ever. Yeah, but if I told you that before, nobody would have listened to what I was talking about. Well, yeah. But what I will definitely ask you questions on is this. Building a transmembrane protein. No, you're never wasting paper when knowledge is there to be attained. It was. 148. All right, so we're going to build a transmembrane protein. This occurs at the rough endoplasmic reticulum. You need a few things here. To start off with, you need a ribosome and RNA. What organelle is going to spit those out, ribosomes and RNA? Yeah, the nucleus. How do they get out of the nucleus? Through what? Through the nuclear pore. So they leave through the nuclear pore. The first stop they have, pardon? Ribosomes and RNA. It's coming out of the nucleus to the nuclear pore and going to the rough endoplasmic reticulum. Now, in the rough endoplasmic reticulum, you've got these C-shaped proteins. They look like this. Well, like C's? Yeah, like C's. Is that C's for you? Yes. Okay, cool. These are sort of, imagine this is the membrane, like I'm a mime today. Here's the membrane. These C-shaped proteins are floating around in it. They're going to match up to each other, forming a tunnel. Okay? The ribosome is going to bind to the top of that tunnel. So the ribosome, now we're shifting the whole thing. Imagine it's sort of rotating on its axis. Now, this is the membrane. Here's the C-shaped protein coming together. The ribosome comes and sits on, there we go. The ribosome sits right on top of it. So far, so good? The ribosome's job is to produce a polypeptide. So the RNA binds to the ribosome, and then it starts to produce a polypeptide that gets pushed into the rough endoplasmic reticulum. The only way it can get in is through that C-shaped channel. So here you've got this polypeptide that's being pushed through, sort of like through a hole.
So the ribosome
Exactly. Now the deal is, though, some of these amino acids are hydrophilic. Some are hydrophobic. The hydrophilic ones go right through and enter into this aqueous area of the endoplasmic reticulum. The hydrophobic ones don't want to leave that channel. They want to stay away from the water. If 20 hydrophobic amino acids build up in there, it creates a clog. It's sort of like when you clog a toilet. You've never clogged a toilet. When your friends clogged a toilet at some other place. And inevitably, it's like at a party or something. The idea is once the toilet's clogged, or once the C-shaped channel is clogged, there's no way you can push more amino acids in, or more associated feces or whatever. What happens when you flush the toilet and it's clogged? Right. Thanks for letting me know. Water flows up and over. Does that make sense? Here, if the amino acid chain is clogged up that channel, the ribosome actually pops off, leaving a chain of amino acids behind it. So now you've got a hydrophilic region on the outside, a hydrophobic region that's stuck, that clog, and a hydrophilic region. Pardon?
Say that last part one more.
Oh, I'm sorry, Olivia, what did you say? Like a zit? I guess if you pop it and then you get stuff coming out, you can think of it that way. So you've got a hydrophilic region down here. You ended up with a clog of about 20 amino acids there, and a hydrophilic region up there.
Wait, so if it clogs the channel, then what does what pops off?
The ribosome. Right. And then that C-shaped channel dissolves, breaks apart. Yes?
How does it go through the bilayer hydrophobic?
Because it's got that channel there, and that channel effectively creates a localized environment for it. So what's happening is you're pushing in amino acids. Some of those amino acids are hydrophobic, so they don't want to get down here. So that's how it gets stuck in that channel. Then once it's done being threaded through, the channel breaks apart, leaving just the protein in there.
So how would you summarize from
Step 2, after about 20 amino acids that are hydrophobic, the ribosome, the channel gets clogged, and the ribosome breaks off the top.
Oh, so the clogging is like a normal process.
Totally normal. It's what they need to happen to get them bedded. No. Yeah, this is how you create an integral membrane protein, a membrane protein that spans the phospholipid bilayer. Any questions? So now we've got the structure of the membrane down. Now we've got to talk about the function, how it works. So all of that chemistry that we just talked about comes into play with transporting molecules across the membrane using diffusion, osmosis, or active transport. Selective permeability is a critical role played by the plasma membrane. It allows certain things into the cell, but not others. It's sort of like your relationships, I assume. You want some things to come in, you want some people in your life, you don't want other people in your life. Especially true now in like social media. You're like, block. If you haven't done that yet, it's totally good. You don't have to see whatever these other people say. It's great. So you want to allow some things in, but not others. Because if you allow too much in, you're going to get toxified. You're going to get a lot of bad material in. If you're going to let too much out, you're going to end up releasing your nutrients. And you don't want to do that. So the cell membrane prevents things from getting, prevents nutrients and substances from getting out that you don't want out, and prevents them from getting in that you don't want in. There's a few ways across with diffusion, facilitated diffusion, and active transport. The key to this whole thing is that hydrophobic center of the phospholipid bilayer. We've got a hydrophilic head, a hydrophobic layer in the center, and then a hydrophilic layer. It's sort of like Tootsie Pops. If you look at a Tootsie Pop, you've got this candy outside, hard candy outside, and this chewy candy in the inside. They have completely different properties. Whereas it's easy to bite down on the center part, it's harder to bite down on the exterior part. So, I've noticed, and maybe I'm wrong, a lot of people may like the outside part, or they like the inside part, but the two may not go well together. Oh, you know what else is like? What are those things? So blow pops. You have a blow pop and you're like, I would like some gum. Well, you can't have it. It's trapped inside of that candy. Well, I just want the candy. Well, I don't want gum. So you've got two totally different layers going on, each with distinct properties. And molecules interact with both distinctly. Some molecules are highly permeable. They go right through. They go right through the membrane as if it didn't exist. Gases and small uncharged particles can fit between the different phospholipids. They're small enough to slide through. It's almost like, I was at an amusement park with my son, and he's small, and I'm bigger than him. Surprise. So he was able to negotiate his way between the crowd and get far, far away from me. I couldn't. I was stopped by the larger particles, the larger people. Small things can get through places that other things can't. In this case, gases do. I'm not sure how much you know about the circulatory system, so I'm going to give you a super brief, here's everything I know about anatomy, because I really don't know much. Anatomy is not my thing. You breathe in oxygen, right? It comes from out here in your lungs. And there's all sorts of ways it gets there. It could go through your nose, but if your nose is stuffed, it goes through your mouth. You can get it there all sorts of ways. Oxygen goes down to your lungs. In your lungs, you've got blood vessels down there that are really close to the surface so that oxygen can go from high concentration in the lungs into the blood cells. So it sort of like sidesteps in. And then the blood cells carry that oxygen. Now, the only reason they were able to move is because there was a low concentration of oxygen in the cells, high concentration in the lungs, and it moved over. And then as those blood cells start moving through your body, because you got this part and your heart pumps things, they're sort of leaving that oxygen behind, almost like a, if you've ever cooked pasta. You've cooked pasta? Yeah, good. All right, you take pasta. It's in a pot. In that pot is pasta and water. You take it over to a strainer. Where's the strainer located? That's the best answer. Not always the one I go with for some stupid reason, but it works. You take it to the sink. You pour the bowl into the strainer and you lift it up. What's coming out of that strainer? Water. Now you got to get the pasta from the strainer to the bowl that you want to put the pasta in. Where's that pasta bowl? Hopefully right next to the sink, right? Sometimes, if you don't plan well, it's like in the cupboard or it's all the way across the room. So you take this thing of pasta with the strainer that's leaking all of this water. You go across the room and you dump it into the bowl. You have now left behind a trail of water. Does that make sense? Not if you're trying to catch the boiling water. That water dripping out is exactly what would happen from the strainer, is exactly what would happen to the red blood cells. Oxygen's constantly streaming out of them, going to the cells around it, until they finally get to their destination where they slow down a bit. And we'll get into that in like 102. We'll talk about capillaries and we'll talk about webbing and how everything slows down. But once it slows down, the oxygen finishes diffusing. It goes to the area of low concentration in the other cells. And it picks up while it's down there carbon dioxide, which then goes back to the lungs. You get these blood cells back up to the lungs, and they push this carbon dioxide from inside of them, where you have a lot of carbon dioxide, to the lungs, where there's a little bit. And it proceeds right across that membrane as if there was no membrane there at all. Super highly permeable. Gases and small uncharged molecules. It goes right across. It's like the membrane doesn't exist. Water's pretty darn permeable. There's a bunch of proteins in the membrane called aquaporins. And what aquaporins do is they are just big enough to allow water molecules through. Not to allow bigger molecules through, just water. They're selective in that way. And when they've got a hydrophilic region that sucks them in, almost like a little water vacuum. They can pull billions of water molecules across a second. So it's as if this membrane was totally permeable to water. Water's just constantly flowing in, and it's constantly flowing out. Only water. And then you get more molecules. They get bigger. They get less permeable. Polar molecules, sugars, they're easily dissolved. They can get in sometimes. They can fit through channel proteins, but it's getting harder and harder to get in for them. Why? Because they're a little bit too large to fit, or they may, when they hit the phospholipid bilayer, they're interacting with that hydrophobic layer and it's excluding them, it's pushing them out. Oh, definitely. Not all molecules are the same size, without a doubt. And these molecules, amino acids, proteins, nucleic acids, polysaccharides, most of our macromolecules, they're too big. They won't fit. So they can't make it through that membrane at all. So you have gases, small uncharged molecules, and water that can very easily cross that barrier. Just about everything else needs help.
So, like proteins and nucleic acids and amino acids can have help in?
They could make it through, but they need help. And we're going to talk about what helps them through. Before we get there, we're going to talk about small molecules moving along what's called their concentration gradient. A gradient is where you have a high concentration in one area and a low concentration in another. Solutes will always move from a high concentration to a low concentration automatically. No energy required, always happening, until they're evenly spread out. That process is called diffusion. If you guys ever, you've had to have, if you've made Kool-Aid or you've made some sort of instant iced tea mix, you put the iced tea in the container or the Kool-Aid in the container, it sinks to the bottom. That is a high concentration at the bottom. You wait for a day or 2 days because you don't want to stir it on your own because that requires energy. Over time, those molecules evenly distribute themselves throughout the entire solution. Thing is, if you were to put a membrane right in the middle, that's where it mucks things up. Before I go on, you guys do remember what a solute is, right? Talk about, sorry. You're like, yes, move on. A solute is a particle that's been mixed with a solvent to create a solution. If I was to take salt and then put it into water, I get a saltwater solution. The solute would be the salt. So we're really talking about solute concentrations. Ooh, and what unit of measurement did we have to measure solute concentration? Moles. That's why we learn moles. But the thing is, it's not just the amount of solute that's present, it could be the charge. You'll notice here we've got an equal number of solute on either side. But on one side, we have more positive charges, on one side we have more negative charges. When you have difference in charges on either side of the membrane, you've created what's called an electrochemical gradient. So you have charges on this side that are positive, charges on that side that are negative. They want to even out. So what you end up having with an electrochemical gradient is a need to move molecules from one point to another to even the whole thing out. These positives need to come over here. Those negatives need to move over there. That creates a flow. And in biological systems, we can use that flow to create energy. This is ultimately the basis of cellular respiration.
So it's the solute could be charged positive or negative, and this occurs from both these like two positive charges.
Right. If one side of the barrier has a lot of positive charge molecules and one side has a lot of negative charge molecules, they want to shift sides. So it's equal. And you can use the energy of that movement to power your biological reactions. these gradients can also move water across a barrier, because it's really hard to move solutes across a barrier. We just went through everything that can't get across the membrane. Water can, though. So in order to balance out solute concentrations, to reach that equilibrium, water can move from one side to the other. So over here we have hypertonic, sorry, words you don't know yet. We have lots of solute. Yes?
Sorry, this is
No, please.
electrochemical gradient. So
Mhm.
when there's a net positive or negative charge on one side but not the other is when that occurs.
Yes, that's exactly when it occurs. When there's a net positive charge on one side and not the other for an electrochemical gradient. So here we can use gradients to move water. Here's our semi-permeable membrane. It'll let some things through but not others. It'll let water through, for instance. On this side, you'll notice there's some molecules that are just too big. They still want to have roughly the same molarity. So what will happen is the water will rush from the side with less solutes to the side with more solutes. Membrane. We can describe these solutions in different ways. If we have two solutions that have equal amounts of molarity, equal amounts of solutes per unit volume, they would be termed isotonic. Iso means same. Isotonic. Isosceles triangles all have the same angles. Isotope, isomer. So iso, we have the same solute concentrations. Hypotonic, hypo means small, a hypodermic needle. Hypoglycemic. You have less solute in solution than you have in the cell. Right, so hypo is small, less solute. If you're less solute in solution than in the cell is hypotonic. Smaller amount of solute. And then we have hypertonic. What does hyper mean? Lots. If you're hyperactive, lots of energy. In this case, there's lots of solute outside the cell, not so much inside the cell. So hypertonic, lots of solute in solution, not so much solute comparatively in the cell. So it could be isotonic. Two solutions could be isotonic to each other. One could be hypotonic to the other, which is hypertonic. Water will always move from hypotonic to hypertonic. That's a statement you're just going to have to remember, and it will solve a lot of your problems. Water moves from hypotonic to hypertonic. Because we can't really move solutes that well. There's a membrane in the way. Here we have a hypertonic solution. Here we have a hypotonic solution. Water is going to move from this hypotonic solution into the hypertonic solution. And it will actually shift these areas. There's a membrane separating them. Always hypo to hyper. Water moves hypo to hyper. That water part's important. Yes, Olivia? Water will always move from hypotonic to hypertonic. Water moves from an area of low solute concentration to high solute concentration. That movement of water is called osmosis. It balances out solute concentration. Osmosis Jones. Yeah, it had nothing to do with osmosis. I watched it just to find out about osmosis. Didn't work. Osmosis is a type of transport that's termed passive because it requires absolutely no energy. There's active transport that requires energy. This is called passive. So here's an example. Water is moving from the solution into the cell. So How would you classify the solution? Hypertonic, hypotonic, or isotonic? Hypo, because water moves from hypo to hyper. Here, what would you classify the solution? Water's moving out of the cell into the solution. Hypertonic. Water always moves from hypo to hyper. And then this one, water's moving evenly in both directions, so it's isotonic. Would I just give you one that had, I don't know, 2 molar on the outside, 1 molar on the inside? Which way would the water go, out or in? 2 molar on the outside, 1 molar on the inside, which means there's a lot of solute on the outside. So the water will flow, the water's going to flow toward the hypertonic solution, the one with the more solute. That's stupid. Sorry. In animal cells do not have a cell wall. So there are 2 consequences of putting cells into solutions that are not isotonic. Animal cells do not do well outside of isotonic solutions. You might have osmotic lysing, or you might have crenation or crenellation. Both are the same. With osmotic lysing, water will move from a hypotonic solution into a hypertonic solution. If you were to take pure water and surround cells with it, the water would flood into the cells, causing them to eventually pop. Sort of like water balloons. When you guys, I assume you've thrown water balloons at people, with or without their knowledge. That's cool. Do you fill them up at the sink, or do you fill them up at the hose? The hose? Okay. If you got a little funnel, there you go. So you take these water balloons, you fill them up with the hose. First type of water balloon you fill up, you only fill it up part way, you tie it off, you throw it at somebody, it bounces off them. And then you've lost the element of surprise. Second type of water balloon is the perfect water balloon. It's filled up just right. You throw it at them, on impact it explodes. The third type of water balloon, though, is the one we've all experienced, which is osmotic lysing. You fill it up, and you think this is going to be the best one ever. It gets bigger and bigger and bigger until what happens? It pops. Because the structure of the outside cannot contain the water on the inside.
So did you say it goes from hypo to hyper?
Water always moves from hypo to hyper. So if you have a hypotonic solution outside and a hypertonic solution on the inside, water floods in.
Wait, could you repeat? Because you were explaining to her a question. Could you repeat?
Yes. So water is going to move from a hypotonic solution to a hypertonic solution. Outside of this red blood cell is a hypotonic solution. Water will flood into the red blood cell. It will fill up like a balloon, and it will eventually pop.
Wait, you just said that hypertonic was on the outside?
Hyper is on the inside.
Okay, I thought
Hypo is on the Sorry, they sound very similar. I'm sorry about that. I'll try to enunciate better.
you're fine.
For crenation, in this case, you've got a hypertonic solution on the outside and a hypotonic solution on the inside. So which way does the water flow? Out. In this case, the cell shrivels up. Yes?
So does water not diffuse evenly? Doesn't seem like it does.
Water diffuses evenly. The problem is these membranes are going to stop these solutes from getting out. So water is going to diffuse in order to make it all even. So
Well, it's about the solutes then, right?
this is about which way the water's moving because of the solutes. Yes? Okay, I'll wait. Okay.
How do you know which is hypertonic and which is hyper?
You will know based on the molarity. The higher molarity is going to have more solutes, and which means it's going to be hypertonic. The one with the highest molarity.
And you said for the second one that the hypertonic solution's on the outside,
Right, so hypertonic's on the outside, so water leaves the cells.
and that's what makes it dry up.
And that's what shrivels it up.
You
Yes.
said highest molarity is
Hyper.
hyper.
What's
Um,
that gonna look like? Because I'm confused
Uh,
by that. Like, how are we gonna know, like, what the higher?
if I give you, I'll either give you numbers that say this is 2 molar solution, this is a 1 molar solution. Which way is the water moving?
Okay, so that would be 2 to 1.
That would be, actually, it'd go from 1 to 2. Which way is the water moving?
So you said
So 1 molar solution would be hypotonic, yeah. Always remember, hypo to hyper for water. Question? Right. Okay, the way you can think about this. Slugs. If you were to take a slug and drop it in a bucket of water, water will flood into the slug, and the slug will blow up like a little sphere of the slug. Go and try it if you want. So water floods in. That slug is acting like a cell. What happens if you put salt on a cell? You create a hypertonic solution around the slug. It dries out, it shrivels up. I assume you've done that. I mean, maybe you're not a horrible person. That's cool. Whatever. I do it to protect my jack-o'-lanterns. Take a ring of salt, put it around my jack-o'-lantern. It stops all the slugs from eating it. What, a ring of salt around it? Oh, good point. Always thinking. Yep. Now, plant cells have a different reaction because plant cells have a cell wall. And that cell wall is made of cellulose. It's made of interlinked carbohydrates that make it really, really rigid. So if you were to take a plant cell and put it in a hypotonic solution, pure water, the plant cells absorb that water and become what's called turgid. Very rigid. They have a lot of pressure. That's what, when a plant is wilty, what do? You give it water. The water goes into the stem, it makes everything more rigid and gives them much more strength. The flip side is if you were to add a hypertonic solution to a plant. Salt water. You guys did that in lab, I believe. Add salt water to a plant, the plasma membrane rips away from the cell wall and puts all the organelles in one corner. It's lost water. It can't survive. It wilts and dies. That's why if you were to, I don't know, try watering plants with Gatorade. I had a student decide to do that one year because, you know, Gatorade's got electrolytes in it. All the plants died across the board because there's too many solutes. The Romans and the Carthaginians, how many of you guys have taken world history or I guess Western Civ? All right, do you remember it was the Pyrrhic Wars, wasn't it? Where the Roman Carthage is in Africa, Rome is in Italy, they fought each other. The Romans beat the Carthaginians back down to Carthage and they're like, all right, you stay there, we're going to go back to Italy and chill and take over the rest of the world. And they're like, okay, that's cool. A few decades later, the Carthaginians were like, hey, let's go mess with Rome. So then they went across the Mediterranean, attacked Rome. Romans beat them back to Carthage, and Romans got there and were like, okay, we said stop. Just stop. Okay, no, we're not going to do anything. Carthaginians then said, okay, we got a new idea. We're going to go and attack Rome, but this time we're going to go up. We're going to take our elephants with us, because that's a great idea. Take our elephants up through Spain, cross through France, across the Alps, down the Pyrenees Mountains, and hit Rome from behind. And they did that, and a lot of their elephants died. But they got to Rome, and then the Romans beat them all the way back down to Carthage. And this time they said, okay, you're done. We're going to salt your fields. So what they did was they took salt crystals, laid them out in the fields, and that salt created a hypertonic solution. So now the plants wouldn't grow. And the plants wouldn't just grow for a year, 2 years, decades and decades. There was famine. The Carthaginians did everything they could to survive, and it was really hard for them. They couldn't make war on Rome anymore. Similarly, my crazy aunt, she decided, she's one of those people who's really into conspiracy theories, and decided that she was going to make her own herbicide. So she made it out of salt. And she decided to spray all of her garden with this salt herbicide to kill the weeds. And she was shocked, utterly shocked, that every single vegetable in her garden also died. And she's like, I made a super herbicide. And I was like, no, you just, literally, thousands of years we've known this would happen. But the Illuminati, so you know. If you were to put a plant into a salt solution, they are going to die because the water gets removed from the cells. If a plant loses 40% water weight, you cannot recover it. For us, if you've been dehydrated, people tell you drink more water. If you go to the hospital and you're dehydrated, they don't hook sterile water up to your arm. What would happen if they put sterile water in your arm? You would scream, ah, they put a needle in me. Why would you die? Why would it be bad? What would happen to your cells? You got pure water going next to these blood cells. They would lyse. Every single one of them starts popping like a little balloon. And it's really cool to watch. Unfortunately, we can't do bloodborne. We can't play with human blood in here because of possible disease. But hypothetically, if you were to look at a drop of blood in a drop of water, what you would see, literally watching under the microscope, is pop, 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 little fireworks going off as all of the blood cells lysed. Water rushes in, causing them all to pop.
Are you able to do like blood cell stuff at like VCU? Or are you just referring to like here?
I think for Bio 101 students, it's not a good idea to do
like
it with.
bioscience.
That would be fine, yeah. It works with animal blood too, yes. What would happen if you put a red blood cell into a hypertonic solution? All the water goes out. You need to make an isotonic solution, which is why they give you, when you're dehydrated, saline. Because what saline is it's has the same level of solute as your blood cells, as your plasma.
Is that why you can taste it when they like flush?
Maybe. I've never tasted in the back of my throat when they did that to me. Animal cells need to protect themselves from this osmotic lysing. The way they can do that is with a contractile vacuole. Vacuoles are important not just for storage, but they're important for maintaining cell volume. What happens is water floods into the cell and it expands. And then after it expands, the cell uses energy to squeeze it shut and push the water out. Contractile vacuole. So that's osmosis. Osmosis is just sort of the natural extension of diffusion. Moving from an area, solutes moving from an area of high concentration to low concentration. So these are two distinct processes, diffusion and osmosis. Osmosis is the movement of water. Diffusion is the movement of solutes. Osmosis, water moves from hypotonic to hypertonic. Diffusion, you're moving from an area of high concentration to an area of low concentration. And it is passive. It happens without any energy. Here, you've got individual molecules of dye. High concentration on the left, low concentration on the right. Over time, they reach equilibrium where they're equally spread apart. So diffusion is the movement of solutes from an area of high concentration to low concentration. Here's an example. Okay, before we go, what color is this? Blue. Okay. Whoever I hear first, that's the color we're defining it as. What color is this? Green. What color is that? Red. Okay, good. And the reason I have to define those is because I once ended up having the class spend 15 minutes arguing over colors. So these blue ones, which way are they going to move? Toward A or B? They're going to move toward B. High concentration here, so they'll expand out over here. How about the green ones? They're not going to really go anywhere. They're already in equilibrium. And the red ones? They're going to move toward A. That's diffusion. Now, most solutes, as we know, cannot get across the membrane all by themselves. They need help. What will help them across are transport proteins. These alter the permeability of the plasma membrane. They make it so that more individual molecules can get through. One type are the channel proteins. These are not very specific. They sort of create big open holes in the membrane. If the molecule is large enough to fit through the hole, it gets in. These molecules moving through channel membranes follow their concentration gradient. On the outside here, that extracellular fluid, you'll notice it's got a higher concentration of solute. That high concentration of solute wants to diffuse to the inside where there's a low concentration of solute. If you want to be more specific, you're going to use a carrier protein. Carrier proteins are going to grab a molecule from one side of the barrier and then selectively pull that one through and drop it off on the other side. There are three kinds of carrier proteins: a uniporter, symporter, and an antiporter. For a uniporter, you are simply moving things from one side of a cell to the other. That's it. Whole thing. I'll show you again. I'm the uniporter from here to there. Questions? Uni means one. I can move one at a time. A symporter. With a symporter, you have two proteins or two molecules moving in the same direction. So here's the membrane. I'm the symporter. I bind to one first. That opens up a binding site on another place. It binds, which allows me to drop both off on the other side. So again, I can bind once. That opens up. I can bind another and then drop both off.
Only one has to bind.
One binds first and then the other binds. You have to have two binding at the same time. Two bound at the same time. That maintains ratios. So if you're making salt, now your cell's not going to make much salt, you would take in a sodium ion and a chlorine ion so that you'd always have the same ratio of sodium to chlorine.
What does it bind?
Bind, hold on to.
Like, what is it binding?
The transporter is going to bind to the whatever it's on the outside that wants to bring in. If I needed to bring this into the cell, I have to bind to this. So it's the
Binds to. She's asking what is that supposed to be?
that's nutrients, ions, anything the cell needs. I'm sorry, thank you. Appreciate you clearing it up.
Part of a cell.
Part of cell food. Yes.
Could you do the first one again?
The uniporter? Sure. With the uniporter, it binds, carries it across. Thanks for letting me use your stuff. A symporter, two things on the same side have to bind together before they're allowed to be brought across. With a symporter, two items from outside the cell have to bind, and they're brought through to the other side.
So we're taking things from outside the cell.
Bringing them in, right. And then an antiporter is where you have two molecules moving in different directions. So for this to work, you bind on one side of the cell. That opens up a binding spot on the inside, and then they're able to switch. So they're moving against each other, anti.
Could you say that again?
Sure. So first, you have a molecule bind on one side. That opens up a binding site on the other side. So now I have one on the inside of the cell, the other's on the outside of the cell. And then after they've both bound, they switch places. Why not just have like two one-way ones? Is that the question?
Or like, why is there 3 different ways of this?
Why is there 3 different ways of getting it through?
Yeah.
So a uniporter is going to constantly, it doesn't matter the amount that's inside, it will just keep bringing it in. With a symporter, you're going to bring in equal ratios of two things. And why that's important is, so if you're making salt, hypothetically, salt's an easy one, NaCl, right? If you're making salt, you don't have 1 sodium and 15 chlorine. Does that make sense? You don't need 15 chlorine, so you only need them in a 1-to-1 ratio. So you bring them in together in that exact ratio, and you never have too much. And then with an antiporter, you can use it in order to, one, get rid of every time you get rid of waste, you're pulling in something new.
So these are the 3 types of carrier proteins, and what are they carrying in again?
They're carrying in ions, they're carrying in nutrients, they're carrying in vitamins, they're carrying really just about any molecule. Yeah, carbohydrates, nucleic, well, not really nucleic acids. They bring in sugars, things like that. No problem. Uniporters, symporters, antiporters. Now, as long as you're following your concentration gradient, that does not require energy. It happens naturally. The problem is when you go against your concentration gradient. That's when you require energy. So we get, thus we beat on boats against the current, borne back ceaselessly into the past. Thank you. That is the Great Gatsby. In case you guys didn't know, that's the Great Gatsby. My first semester teaching, I made the mistake of saying, hey, if you know what that quote's from, I would give you an A in the course. And then I realized, oh, that'd be a horrible thing, because then I just have to give Amber an A. So I stopped doing that. But you do have my sincere appreciation. The Great Gatsby was a wonderful book, and great life lessons there. Don't look back into the past. Don't reach out for the green light at the end of the dock. The big thing is, boats against the current. If you are in a canoe and you're on a river, it takes barely any energy to move forward with the current. It takes a lot of energy to move against the current. If you've ever been in a crowded city, and I don't know that Richmond's that terribly crowded of a city. If you've ever been in like New York trying to walk against the crowd, it requires energy. Going with the crowd doesn't.
So I'm confused, like, how does that relate to what we were just talking about?
How does active transport or the...
So
So we were just talking about those transport proteins, right?
So are you saying which ones of these is, like, easiest to do?
If that transport protein is moving with a concentration gradient, it does not require energy.
Okay.
If, though, you're pumping against it, if you're using a uniporter, for instance, to push a molecule from an area of low concentration to high concentration, that's going to require energy. They naturally follow that concentration gradient, but you're making them go against it. So that needs energy. That needs ATP. Any of them will if they're going against the concentration gradient. Anytime you move from an area, you're trying to push solutes from an area of low concentration to high concentration, you need energy to do it.
So that is, we're looking at diffusion there, not osmosis.
Correct. If we were looking at osmosis, it would be the movement of water, and these are not water. These are not water molecules. Yeah.
Osmosis used when we're talking about the phospholipids membrane?
We're talking about the membrane, and we're talking about going directly across it using water.
The water is just one aspect of it.
Yes.
We also use different solutes and things.
Right. So now we're talking about a different way of. So we talked about getting water across using osmosis.
But is osmosis and diffusion happening at the same time?
At the exact same time. You have a lot of processes happening at the same time. Sometimes we can use a pre-existing gradient to power a pump. We can use the power of a gradient to provide energy for our pumps. This is called secondary active transport. This is sort of like if you're a jerk, hypothetically. You guys have used revolving doors before?
Yeah.
How does a revolving door work? You don't use it. So you go counterclockwise. You go to this revolving door and you push on it. That pushes the door around and you can go through, right? But you don't have to actually push on it. How can you get through it without pushing on it, without expending any energy? You stand and, just like Isaac said, you stand and you wait, because what's going to happen eventually? That's right. Somebody's going to come from the other direction. And if they come the other direction, they're going to push on that, and that lets you get through without you expending any energy. So it's passive on your part. Same thing happens with molecules. You can move from an area of high concentration. You have some molecules pushing through, and as they push through, like with say an antiporter, they're pushing in one direction, so another molecule can get pulled through in a different direction without the use of energy, because you're using the energy of the other gradient.
Can it ever just go too fast and nothing gets in?
So nothing gets in? I don't think so. I think these things move very, very efficiently. Now, if you combine this with ions, so you have more ions on one side of a membrane than on the other, you've got an electrochemical gradient. You're using that spinning action in order to provide energy to power reactions. Literally, you would have water wheels here that are spinning and creating energy. So you can form electrochemical gradients. Those gradients can then power the creation of what are termed energy intermediary molecules. This becomes the basis of aerobic cellular respiration, which we'll get into in a few weeks. So it's not terribly important for now. It will be terribly important later. Some molecules are just too big to move through channels, too big to fail. Nucleic acids are huge. Certain proteins are gigantic. In order to move those through the membrane, you have to use a process called either exocytosis or endocytosis. Exo means out, cyto is cell, and osis means action, so outside cell action. What happens is we've got, for exocytosis, we're packing things up in the Golgi apparatus for secretion, individual molecules. Those are being sent in a vesicle over to the membrane, where they're released to the outside. Endocytosis works in the exact opposite way. A particle binds or gets close to the membrane distorts, actin filaments slide along, and you end up engulfing it in a vacuole. So you take in a vacuole. So what you can do, you could be packed up in the Golgi apparatus. You go out, you hit the membrane, it fuses, and then opens up and pushes it out. The other way it goes, you could have it come in and bind, a vesicle forms around it and pulls it in. So that's exocytosis and endocytosis. Exocytosis and endocytosis are big, big means of cell signaling. The membrane of the cell is the primary interface between the cell and its environment. Without the cell membrane, or without the proteins on the cell membrane, you won't be able to understand what's happening around the cell. It's like being in a submarine without being able to see outside of it at all. So the proteins on the membrane are key for signaling between cells. There are 5 ways I want you guys to understand about signaling in cells. Now, signaling is important because without signals, your cells would not be able to have coordinated responses. It's like each is on their own. You could have dissolved molecules passing through junctions, direct contact between membrane-bound proteins, paracrine signaling, synaptic signaling, or endocrine signaling. We're going to go through each individually. So first, the local ones. Your cells can signal each other through junctions. Remember desmosomes, gap junctions. You can get molecules from one cell into the next. But they have to be cells that are right next to each other, thus local. So this is direct sending of molecules between the cytosol. Yeah, junctions are where they meet. So you have gap junctions, desmosomes, and tight junctions. Tight junctions are sort of rivets. They hold it together. Those gap junctions allow for cytoplasm to flow evenly and smoothly. Signals can be sent from proteins in the membrane to membrane contact. If you put a cell, it's like the cell is putting out little flags, and the cell next to it is close enough to read the flags. So their membranes are interacting. You might have what's called paracrine signaling, which is very, very similar to synaptic signaling. With paracrine signaling, you've got a cell secreting molecules through the membrane. So the Golgi apparatus is packing up these molecules, sending them out to the membrane, and then releasing them. Those molecules can then bind to receptors on the other cells. This is also known as the mediated cell response, receptor-mediated response.
These are the 5 ways for
Cells to signal, yeah. So this doesn't, though, travel very far. It only really affects the cells surrounding that cell. Synaptic signaling is very, very similar to paracrine signaling. The difference is you're using neurotransmitters that are created in the Golgi apparatus. For those of you who have taken psychology before, these are probably familiar. So what will happen is you're going to have neurotransmitters created in the Golgi. They're packed down, and they're sent out to an immediately adjacent cell that will then absorb those transmitters, or those transmitters will bind to the outside of the cell, causing a chain reaction inside that second cell. So those 4 types of communication are local. They do not affect cells away from this originating cell. It's like when you're yelling, you can only yell so far. Which is creepy now that I say it, but you know. Yeah. Everything up to this is local. Yes, junction, passing cell information directly across cytoplasm. The long-distance signal is endocrine signaling. You have hormones, and people are like, oh, teenagers, their hormones control their life. The thing is, everybody at every age has hormones controlling their life. Hormones are the way your cells signal long distance. How your the pituitary gland signals to the rest of your body. How your hormones move from one part of your body to the other. Now remember, hormones are steroids. They are lipids that are not very water soluble. So if you put them in your bloodstream, they can travel a long way. Following your bloodstream, they can then cause cells in other areas of your body to react. One part of your body may send a signal that says one thing, and then it gets to another cell at a different part of your body altogether, and that will cause a cascade to happen within that cell. That may need to lead to local signaling of the other cells around it. So those are 5 big ways of signaling. Endocrine signaling. Long distance. This is the only long distance one. I am concerned about you guys knowing this semester. So that's how we get the signal out. Now we need to have a reaction of the cell to the signal. So you're going to have a reception, transduction, and response. 3 stages. You've got to receive the signal, change the signal a little, and then respond to the signal. So step 1 is reception. You get a signal molecule that a cell recognizes. Maybe it binds to a receptor, and the cell's then able to say, hey, there's a signal and it means something. So you receive the signal. Step 2, you transduce the signal. It's slightly modified, so it causes a chemical reaction to happen. Here we've got a signal coming in and binding to a receptor. So that's reception. Then something's going to happen in here that's going to produce messages inside of the cell. Any of you guys ever been on a phone tree at some point in your life where one person sends out, of course that's not a thing anymore, is it? Because you have a mass, never mind. Okay. Have you ever gotten a signal from somebody that caused you to do a whole bunch of different actions? I don't know. You got a call from your parents that say you have to babysit your brothers and sisters. If that happens, you get the signal. The signal is, you receive the signal from your parents. That signal was, go take care of your brothers and sisters. That causes several more things to happen. You need to start preparing to do things. Start preparing dinner. Start, I don't know, making sure they don't cry because now you're in charge. All sorts of things. You've got to change the signal into something that the cell understands. And that leads to a final response, what the cell actually does. So you get the signal, reception, transduction, changing the signal into something the cell understands, and then a response from the cell that then could lead to more signals being sent to other cells and more things happening. That's called a signal cascade, but we're not getting into that. Now, do you have any questions? That's the membrane, cell signaling, and transport across the membrane. In the super awesome next lecture, we're probably going to talk about, I'd say enzymes and energy and metabolism. Questions? No? Wow, there are a lot of content review questions this time, isn't there? So many. 3 pages worth. 3 slides worth, yeah.